Welcome everyone to the Print Program Proposers Day. I'm Dr. Ryan Spitler, the Program Manager for Print. We're really excited to have you join us today to learn more about the program, how to work with us, and most importantly, how we can all work together. To start things off, I'd like to welcome our ARPA-H Health Science Futures Mission Office Director, Dr. Amy Jenkins, to give you an overview about ARPA-H. Amy, please take it away.
Really excited to be here today and welcome you to the ARPA-H family. Explain a little bit more about the model. Some folks in the audience may have known nothing about ARPA-H. Some folks in the audience may know something about DARPA, which is similar to ARPA-H. And some folks in the audience, I know for certain, know a lot about DARPA. So ARPA-H is modeled after DARPA. And for those of you who don't know, DARPA stands for the Defense Advanced Research Projects Agency. This was an agency that was stood up in 1958 by President Eisenhower at the time, and it was in response to Sputnik. So there was a satellite put up over our heads. People panicked and said, what is this thing that's looking down on us? And the US government said, we will never be surprised again. We are going to stand up an agency that is going to create surprise. We're going to fund the craziest science, and we're going to ensure that this never happens to us again. We're going to be the surprise makers in the world. And that was the model that DARPA was stood up for national security. That led to many innovations. It led to stealth, largely credited with inventing the at least very early underpinnings of the internet. It led to self-driving cars, and it also led to some of the advances that gave us mRNA vaccines during COVID-19. And so with that, in that model, ARPA-H was stood up. But ARPA-H has one mission. It is not a national security mission. It's a health mission. So we are here to accelerate better health outcomes for everyone. So President Biden, in March of 2022, said, I want to stand up an agency like DARPA, but I want it to be able to focus on health. Imagine what we could do if we take that crazy, transformative, wild science agency and we apply it 100% focus on health. And that's what ARPA-H is. So we are modeled after the business practices of DARPA, and you'll hear that invoked a lot. A lot of people, if you are familiar with ARPA-H and its inception and how we stood it up, a lot of people ask the question, well, where does ARPA-H sit in the big ecosystem that is the government? And so this is why I like to present this slide, because there's a lot of confusion about this. So we are administratively linked to the NIH. We are not an NIH IC. We are not an NIH agency. And so we are an independent agency that is administratively linked to NIH, but we are a separate division within the Department of Health and Human Services. Our agency director, Dr. Renee Wegersen, reports directly to Secretary Becerra within Health and Human Services. So she does not report to the NIH director. I say all this to say that our model is quite a bit different. So many of you in the room are likely very familiar with the NIH model, with study sections, with R01s. We don't do any of that. We'll talk through a little bit of our model, but we don't have study sections. We don't fund science by consensus. And the reason that we don't is because there's an absolute place for science by consensus and for study sections like at the NIH. However, here we want to do things that are risky and crazy. And if you put every one of you in the room and you ask you all to come to consensus on something that's crazy science, nobody will agree. We'll never get there. And what we'll do is we'll get to the point where it's not risky anymore. And we don't want to do that. We want to fund things that are crazy, maybe impossible, but that's the point of our agency. So with that, we have 4 mission offices at ARPA-H. I'm not going to talk through all 4 of them, other than to say there's the Health Science Futures Office. That is the office that I am the director of. There are 3 other offices, our Scalable Solutions, Proactive Health, and Resilient Systems Office, and they have the same exact structure as we have. So a director and program managers that are launching programs inside those offices as well. So what does our ecosystem look like? We work very closely with the NIH. We are, like I said, not one of the divisions within the NIH, but we partner with them and work very closely with them. We work with our federal partners at the Center for Medicaid Services and the FDA, and we work with private sector. But where do we fund? You're all here to know who we fund, who can be funded by ARPA-H. We call our grantees performers. That's because we don't issue grants. You're going to hear a little bit about that later. But I get this question all the time, like, I'm a small company. Can I apply to a program? Yes. I'm a large company. I'm a pharmaceutical company. Can I apply? Yes. I'm a small lab. I'm a big lab. All of the above. So we fund academia, industry, NGOs. We work with other government partners. All of the above. And the reason we do this is because the things we're asking you all to do are crazy and they're hard, and we need everybody to come together to the table to try to solve these problems. I'm going to talk just very briefly about the Health Science Futures Office within ARPA-H. So I've told you about ARPA-H and how we fund revolutionary science, how we fund high-risk science. What do we do specifically within Health Science Futures, one of the divisions within the agency? The reason we broke these offices down the way we did is that this is focused on really the types of technology that we may fund. And so when Health Science Futures within this office, this is the probably craziest of the offices at ARPA-H. I like to say that, where ARPA-H is ARPA office. And so here is where we are really looking to expand what's technically possible. We're looking to develop tools, technologies, platforms that will really revolutionize healthcare of the future. And many of the things that we invest in and some of the technologies that we invest in, they may not have that healthcare impact for a decade. But we feel that making these pivotal investments now will entirely revolutionize the future. We have a lot of different methods that we use to fund groups. But one of the things, and I said this briefly before, and it's the reason you're all here today and listening online, is the reason we're going to have those poster sessions. And I love that we had our impromptu introduction to each other. We are funding very hard things. We're asking you to do very hard things. It is very unlikely that any one group out there can do this with one PI, with one lab, with one company. It's very hard. And so we're looking for you to find each other, help each other. You should have complementary technologies that will be applied to solve this problem. That's the point of a Proposers Day. The reason you're here is to talk to each other, find each other, and figure out how to solve this big problem that we're asking you to come together for. And so with that, I just want to briefly talk about a couple of our other programs, give this a little bit of a flavor of what we do in this office. Nitro is an osteoarthritis program, Psi is a surgery program, and Apex is a vaccine program. So we run the gamut. We have lots of different types of technologies that we invest in this office. And briefly, I just want to talk a little bit about our model. I mentioned that we don't do study sections. It is a very different model, and our program managers come here. We come here as government employees, but we don't stay. We're not allowed to stay at this agency forever. We want new, crazy ideas constantly coming into the agency. And because of that, program managers have a very limited lifetime. They're here on a 3-year term, renewable for up to an additional 3 years, so a maximum of 6 years. And then they have to go, I have to go, our director has to go, everybody leaves. So what the agency looks like today, it will not look this way in 6 years. And that is because if you have the same people thinking of the same ideas, those ideas are nobody's going to have, new ideas for the entire rest of their life. We really want to ensure that we bring revolutionary ideas into this agency. So program managers will come from anywhere between 3 and 6 years. They'll start programs while they're here. And those programs run anywhere from 2 to 4, and the slide says 2 to 4, right? It's 3 to 5, 3 to 5 years, and then they go. And so that just keeps, entirely new ideas always coming into our agency. And so how does this work? Who makes the decision of what we're going to fund? Our program managers do. So we're a bottom-up agency. I don't make the decision. Our director does not make the decision on what kinds of programs to go after. We bring in program managers that are very passionate about a space. We said, yes, let's take a shot on this. And so the program managers come in with the idea. They say, this is a challenge in the healthcare field that I want to try to solve. They develop a program. And so where you are right now is you're in that third box, launching the program. So the manager goes out, holds proposers days, folks propose to the solicitation, the call for proposals. And then out of that, groups will be funded. Those are our performers. So that's our general model. And none of that is done through a study section. So this is done through a valid government review, but it's not by consensus. One thing I do want to say is that what you all are trying to do is very hard. And so you may not always succeed. Sometimes some of the approaches that folks take may not work, and we recognize that. So there will be successful failures, if you will. And we really want to push as hard as possible to ensure those successful failures. And then if we manage to get to the very end and are successful, then that's the biggest prize. You're going to hear a little bit later, very briefly, about what happens if you are successful. We don't just, we do walk away. ARPA-H doesn't fund things in perpetuity. There's no renewal of the grant, but we ensure we have a whole office that's focused on ensuring that when we're done here, when Health Science Futures is done, that there's a life beyond the program. And so with that, we like to put this up. This is an installation at the Renwick Gallery in DC, and it kind of summarizes ARPA-H quite nicely. And I want to welcome you to the ARPA-H family. You're part of that now that you've attended an ARPA-H Proposers Day. Think big, think bold, think crazy, say things that you think your colleagues are going to say, person's crazy. I don't think we could do that. That's what we want. That's what we're looking for here in this room. With that, we're going to move on to the next presentation. So thank you all.
Okay, well, hopefully that was a helpful overview of the ARPA-H workflow from Amy Jenkins. And now I'll introduce the PRIT program, Personalized Regenerative Immunocompetent Nanotechnology Tissue. So we all know why we're here, and we're really here to address the organ transplantation crisis. So as we sit here, 17 deaths occur daily, and the lifetime costs can range from 600 all the way up to $1 million. And oftentimes people don't realize that the 5-year rejection rate, depending on your organ type, can actually be up to 60%. And so while this program, we're not going to be able to solve every challenges that we encounter, we will be able to really work on the supply problem of this. And so what we're really looking at was, can we get rid of some of these numbers that are staggering of, we're transplanting 45,000 transplants, but really we need 120,000. And that 120,000 doesn't represent everybody, it just represents what's on our list, which we try to keep as short as possible. So the problem is much larger than this. And this has never been an equitable process, depending on where you live, how far you are from a transplantation center. These are all attributes that can dictate whether you're on a wait list, whether you can receive an organ, and will kind of carry what the likely outcomes can be as well. So beyond the actual supply problem, we also want to work on the immunosuppressive drugs issue. And so we want to make something that we can also have that's immunocompatible with everyone, ideally. Because immunosuppressive drugs have a lot of their own side effects, which can lead to comorbidities. They can be very costly, can be tens of thousands of dollars per month, depending on each individual case. But it's necessary to confer, non-rejection. And this is something that also people don't realize oftentimes is that, having a transplant isn't really a curative event. So the mean lifetime survival of these transplants is about 20 years. So that means depending on how you live, how long you live, especially if you're a pediatrics patient, it can be a very long time. You might actually require another organ in order to, sustain life if you live long enough. And so this is a huge problem that we really want to work to address. And so the way we're looking at this is, can we use state-of-the-art bioprinting technology and patient-matched cells to be able to bioprint organs on demand? So using immunocompatible cells and advanced manufacturing techniques, and then being able to biofabricate either the liver, heart, or the kidney. And we think that this is, a solution that we may be able to really address these huge wait lists that we're seeing, while also getting rid of this immunosuppressive drugs. So if we can match everyone, you no longer need these drugs. And that in itself will be a huge accomplishment to be able to really improve the quality and length of life of these patients, as well as access as well. And so this program, we broke it into 3 different TAs. We want to be able to generate all the necessary organ cell types. We need to be able to use large scale manufacturing to create tens of billions of cells and all the way up to GMP grade and be able to biobank these cells. And then we need to be able to biofabricate with 3D printing systems, have the software hardware necessary to prescribe the necessary structure to be able to recapitulate as close as we can to a native organ. And one thing that's of note is we're looking to restore normal function. We're not necessarily looking to print the whole organ. So this means you could have a partial transplant You could have an ectopic transplant, or you could consider printing, as much or all of the organ if you wanted to. But really what we're trying to achieve is something around at least 40% function to be able to confer survival and a relatively normal patient life and duration. And the other thing is, as you'll see as we go through these technical areas, is that we are offering quite a bit of leeway and flexibility of, being able to put the best solutions forward. And we try to be less prescriptive and allow you that flexibility to be able to do that. And I'll show you how as we work through this. And the other issue, the other item to consider here is also that each team will be required to address all TAs. So we're expecting that you will be basically go after one organ system. So we'll have organ teams, either kidney, heart, or liver. We don't want to see proposals that have multiple organs in one. So pick the one that you think is best and really go after that, ideally. And we understand in terms of this teaming component that there may be industry, it may be academia, it may be a combination. And so we're not necessarily prescribing the teams. We're prescribing basically pick the team that you think is most likely to be achieved this solution. And that's really what we want to see here. So if we look into TA1, generate all the necessary organ cell types. You notice I didn't say generate all the organ cell types. I said all the necessary. And that's because you may not need to, especially if you're doing a partial transplant. You pick the ones you think are necessary to be able to confer the function that you think is achievable within the scope of this program. And you could take the cells, that could be a blood draw biopsy, or it could be biobanked cells. And the challenges in the field, excuse me, have been basically, there's, things like cardiomyocytes that tend to be naturally not proliferative. You can imagine biopsies. Sometimes we have things that are cells are not multi, they lose some of their multipotent potential. But in all cases, the issue has been that, we're just, one of the major challenges in the field has been that we just don't have enough cells to be able to even try in most cases. And so when we think about which cell source to choose, it's really up to each team. It could be autologous, it could be allogeneic. If it's allogeneic, it should be hypoimmunogenic. Basically, it needs to be immune compatible, and you need to be able to create protocols to generate all the necessary cell types, and then be able to look at these cells and, do they look and act? So you have the molecular markers, morphology, and the quality control, quality assurance to make sure that the cells are safe and are behaving the way you would expect them to, as similar as possible to a native cell of that organ type. And so if we look at some of the metrics in the first year, we really want to be able to do, so you'll decide on your solution, one of the ones I mentioned. We're looking at, one of the other issues is beyond accessibility and related to accessibility is being able to keep it cost effective. And so it should be about 5,000 per billion cells initially, with the ability to scale up to 10 billion cells per organ and have a 85% viability. We want to see a high purity, about 90%. And we also want to demonstrate normal function. So looking at having to look and have the molecular markers, within about 20% of the cell type you're driving for. And so within the first year, we're looking to have, we should have the protocol, you should have the cells selected and the protocol identified to be able to do this. And this dotted line basically shows kind of an interphase into 2 and 3, so that these methods can then be used into take into consideration we go into the manufacturing step as part of TA2. And similarly, in terms of being able to get there, because it's very expensive and there's maybe at least 50 parameters you might need to optimize to get this, it's been a challenge in the field to actually be able to produce the kind of cell numbers that we need, and have the partners being willing to take these on, because unless you really know what tissue you're working with, what's the end goal, and all the steps in between, you're going to be spending a lot of time and investment on this optimization step that if it doesn't pan out, it could tank your business. or your research program potentially. So we're trying to take on the risk so that we can get over this hump for the field. And the way we're going to do that is we're going to be developing a cell master bank that's up to GMP grade. We're going to be developing GLP and GMP manufacturing processes so that we have the materials to try both in small and large animal studies. And then also do the same QA, QC that we did in the smaller batches of just the protocol development, but have that, make sure that, we have the same integrity up into the next scale. And of course, we want to make sure that these are patient matched along the way as well. So if we look at the TA2 metrics, so we're looking, the bulk of the work is coming in, year 2 and year 3. And so we'll initially look at GLP manufacturing, about 1 billion cells per organ, looking at about 15,000 per billion cells. You're going to be demonstrating that you can create all the cells you need and at that scale, and also have the biobanking component. Going into year 4, you'll have GMP grade and biobank cells, and obviously a GMP grade is a much higher order, and because of that, for 10 billion cells, it's going to cost about 50,000 per billion cells. And the idea here is to kind of start packaging this up to basically so we have pre-IND enabling studies and documentation. We're not going to actually file an IND at the end of this because it's too early stage to be thinking about that yet, but we're trying to get it so that the normal mechanisms can then take over in the future and be able to address this. And then TA3 is all about organ biofabrication and testing. So the field has been, has constantly struggled with being able to print thick vascularized tissue and has never really been able to recapitulate organ level function and structure. So what we can do now is there's a lot of methods where we're able to print thicker vascularized tissues in that we can print in the vasculature at the time of print and have active perfusion. And that allows us to develop these much more complex tissues. And there's also, we can adjust a lot of the different bioinks that come up that are available to us to think about creating things that have biomechanical properties that are very similar, or as similar as possible to a normal healthy organ. So in this one, we'll be developing a 3D bioprinting system or a hybrid system or whatever is, that you think is the best solution here. But we'll be using 3D bioprinting and also a perfusion chamber that'll allow for active perfusion as well as for maturation development of the tissues as well. And so there'll be, software and a hardware component. We're anticipating that software will be needed for some of the modeling in terms of, vascular branching, or maybe even for the types of mechanical properties that we're looking for. We'll look at different bioink formulations. And the other thing is we're not favoring a particular printing method. You can use any one or multiple combination of methods. It's more about being able to produce the end goal. So there's a lot of flexibility here in terms of the innovation side, as long as you're staying, sticking to the milestones and moving in the right direction that we're asking for, essentially. And in this one, we'll be testing. Initially, we'll have a transition step where we're looking at a humanized mouse. On this way, we can look at structure function of your print, as well as confer immune compatibility. And in the later step, we'll be looking at a humanized pig model, where we'll actually be looking at, can we keep this pig alive? And again, it'll be looking at structure, function, viability, and basically, can we sustain life in an organism that's very similar to the human physiology? So if you look at the TAs, one of the things you'll notice is, we can start developing a lot of this software and even some of the hardware in year 1. And certainly for any of these milestones, you're welcome to, be ahead if you would like to be, but you can't be behind, ideally. So what we really want to make sure is the spirit of this was essentially to make sure that we're kind of co-developing some of these things so that we don't go, oh, wow, I really wish I would have thought about that bioink formulation when I was designing that print head, because now it's clogging or it's too viscous or it's whatever. So kind of thinking about these things up front so that we're not doing everything sequentially in a way that we hurt ourselves in the long run. So for instance, in the first year, if we have a 50 micron resolution in the software, we should also be hitting that 50 micro resolution in the hardware, and it's actually has potential to be able to do that. We like the maturation time to be about 30 days, and then for any kind of bio inks that are be used, we want to see compatibility here. And so we want to see that. So again, as I mentioned, we want to have a bioreactor with active perfusion. We will have the modeling software, and then year 2. We'll really be looking at kind of making sure that we have the complementary, and you're probably co-developing the hardware with this so that you're able to print in the vasculature at the time of print. We want to see that you can perfuse this. And in terms of the characterization, we're going to be looking for the structure function ex vivo and actually in vivo in the small mouse model as well as the larger pig model as we go. But we won't hit the pig yet until after year 3. So year 3, we'll look at testing this in the mouse model with about 10 animals and look at if we can see survival. And then at this point, we know what our solutions are. We have some preliminary data, and we'll have an interactive meeting with the FDA to make sure that we're developing solutions that they're willing to consider in the future to make sure this is going to be something that's moving into translation into the clinic. This is also the primary down select point for teams. So the teams will be computing, basic organ teams, organ specific, as well as between different organs. And those that show the best performance will then be advanced onto the phase 2, which is all about prepping and being able to characterize the humanized pig model. So we want to see that you're testing it in 5 of the pigs. That we're seeing ideally 6 months of survival. We're not seeing toxicity. We're not seeing immune reaction. And we're doing the characterizations to show that this is functioning as much like a normal organ as possible. And then in year 5, we'll do some of the post-mortem characterization. And again, as I mentioned, basically we'll be packaging all our assets up so that we have enough for like a pre-IND filing, and so that this can be transitioned successfully in the future. And so if we look at the print timeline, basically you'll see that, as I mentioned, there's a lot of things being kind of co-developed at the same time. So this is kind of an iterative process of some overlapping components to really be able to stack the deck in our favor that we're developing something that's going to work in the end. Additionally, I'll bring your attention to, we want to see independent validation, in particular for the cell steps. So we want to make sure that a third party can, test the quality function of the cells that you're creating and ensure that, that in someone else's hands, they're getting the same results that you're going to be putting forward as part of your progress within this program. And so, as I mentioned, equity has always been a huge part of what we've been going for. And our goal is to make something that's cost effective and accessible to all Americans. And the other aspect of this is, we're going to really be going for, improving across multiple different fronts, including like if we can do functional kidneys or even improve the current function, can we get patients off of dialysis, which is a huge burden and cost to the US every year. And so this is something that, we're looking to also be, the first time to actually have this be an equitable process, especially for a lot of, in the ethnic minorities who tend to have the most need, have the least access to these. And so by creating either a universal line or creating other methods of creating immunocompatible cells, this will be the first time where everybody potentially should have access to this life-saving organs. And as I mentioned, we will definitely be interested in, make sure to put energy into your commercialization and transition scope of your proposal. So we have a number of different resources that we can tap into, and you'll hear some from the patio team later about how they can help with some of these transition activities and different resources that we can provide. So for those selectable teams, you'll work closely with me and we will work together to try and make this a reality. And as I said, for value creation, we'll generate a bunch of assets throughout this program, and there's a variety of different transition paths, and likely multiple will occur at the same time. We could consider this being transitioned after the ARPA-H funding to a government agency that has good track record in transitioning things like BARDA or DARPA. We could spin out IP into independent ventures, or we consider licensing to either existing companies that may be part of this already, or could be external as well. So there's a lot of potential paths. I think that the main goal here is that this doesn't become dead in the water after the end of the ARPA-H funding period. And we don't expect that to be the case, and we're going to ensure that that's not the case through all our hard efforts that we'll be working on together. So as I mentioned, the impact of print has the potential to be one of the most impactful programs to date from ARPA-H, if successful. We're looking at, can we get close to this 120,000 patients a year need? And thinking about this also at a global level, can we, if we can get this to work as a US process, globally, this is a huge problem as well. So the potential to expand this and to impact the world is huge. And I'm really looking forward to taking this journey with those of us that, we end up working together, and certainly for everyone exploring this together. And the other issue is, really being able to address this issue of immunocompatibility and reducing costs significantly. If we can wipe out the need for immunosuppressive drugs, it's a huge reduce in burden and cost for our patients and our health ecosystem. And beyond this, for the future, this huge impact for the biomedical community as well as in regenerative medicine, because a lot of the discoveries here will lead to advances in multiple related areas and have a huge potential for impact. So looking forward to building this together as a community. And with that, I will then hand it over to Georgina Perry to go over ARPA-H contracting. So thank you for joining us, and look forward to continuing the discussions.
My name is Georgina Perry. I'm the Agreement Officer for the print program. The purpose of this portion of the presentation is to provide an overview of the contractual side of the Innovative Solutions opening and why this method of solicitation was selected for the print program. It will hopefully help answer some of the questions you may have as you're preparing to draft the solution summary and subsequent full proposal if an invitation is selected. Broad agency announcements, also known as BAAs, are a widely utilized and highly recognized process to procure R&D within the federal government. Those that are familiar with DARPA and ARPA will find that the process looks similar. The BAA process is defined under FAR Part 35. The terminology and nomenclature that is commonly associated with the FAR will be utilized throughout the ISO process. However, ARPA-H does not intend to negotiate FAR-based procurements. The reason being is that ARPA-H is seeking an opportunity to invite and award solutions with a portion of industry that has typically shied away from working with the federal government due to the often viewed heavily restrictive terms and conditions. The print solicitation allows for merit-based awards. The ISO allows an opportunity for the award selection to be merit-based and for technical quality to be weighed more favorably than price, which is one of the reasons why a budget range or ceiling is not established. The ISO also allows for best ideas to be weighed more favorably than best price. The ISO will follow a competitive process where the best ideas will be selected and move forward for the abstract process, from the abstract process, also known as the solution summary, to the full proposal process, while each proposer will have a unique statement of work that aligns to the proposed solution. The ISO also allows for a variety of approaches to a problem, not one singular solution. ARPA-H recognizes that multiple solutions are available to one problem, which is another reason why the traditional FAR process will not be utilized. The proposals will not be compared to one another, and each will be evaluated based on their own merit and proposed idea. The ISO is flexible and allows for a variety of awards. The instruments that will be considered and negotiated under this ISO are financial assistance awards and other transaction agreements that will have an opportunity to be negotiated in regards to the terms and conditions, depending on the proposed solution and award type. Some of the ISO basics, award types. The government will allow proposers to propose an award type, but ultimately the best fit for the proposed solution would be awarded. The two award types are, as previously stated, which is the cooperative agreement and other transaction agreements. The award timeline. Full proposal evaluations are expected to be completed by October of 2024, and negotiations are expected to begin in November 2024. During this time, negotiations will include the award instruments, statement of work, and the terms and conditions. Award funding. As previously mentioned, quality and merit are more important than price, therefore no ceiling or budget range will be established. All reasonable proposals will be evaluated and considered. Traditional awards will not be considered, and this includes grants as well. First award vehicle is the cooperative agreement. Cooperative agreements are a form of a financial assistance instrument that carries out a public purpose and is authorized by U.S. law, rather than to acquire supplies and services for the benefit of the government. Cooperative agreements fall under 2 CFR Part 200, 45 CFR Part 75, and cooperative agreements also follow the HHS policy. What differs a cooperative agreement from a FAR procurement contract is the level of substantial involvement that will occur throughout the life of the agreements. There is an expectation that the proposer in ARPA-H will communicate and collaborate frequently to achieve the agreed upon results and outcomes. Because substantial involvement is required for the success of the program, once again, grants will not be considered. In the grant world, substantial involvement is not anticipated. Other transactions. ARPA-H has authority to award other transactions when such use is essential in promoting the success of the project. OTs are a form of a contract with an expressed offer, acceptance, adequate consideration, capacity, and legality. Other transactions reflect commercial contracting practices rather than traditional FAR-based procurement contracts. Other transactions also include substantial involvement from the federal agency, and this will be ongoing throughout the life of the project. Both parties are responsible for achieving the agreed upon outcomes, progress, and results. Some of the pros and cons of another transaction. Pros of another transaction is that many of the laws and regulations do not apply, such as the Competition and Contracting Act, the BI-DOIL, 45 CFR 75, the FAR, the HISAR, cost accounting standards, bid protest, and another transaction invokes commercial practices allowing for negotiating terms and conditions, including the negotiation of intellectual property. Some of the cons include the lack of guardrails that performers typically see under federal assistance or FAR contracts. And another con is that it requires careful negotiation by sophisticated parties within the commercial environments. Some of the pros and cons of a cooperative agreement. A cooperative agreement is a familiar award vehicle and is commonly utilized in awards for academia and those that frequently negotiate and award contracts with the federal governments. Many organizations have designed their systems around these financial assistance vehicle, and there seems to be consistency amongst organizations and their award portfolios. Some of the cons of a cooperative agreement is that the terms are largely set by laws and federal regulations, which limits the ability to negotiate. It includes requirements that may be unappealing to commercial entities. Some examples include profit or a ceiling of unaudited indirect rates. Cooperative agreements are also considered more bureaucratic because, once again, they do largely follow those laws and federal regulations. The process overview. Solution summary. Submission is mandatory. The solution summary should not exceed 5 pages. This excludes the cover page and the ROM. The solution summary should cover the sections outlined in the ISO, and all solution summaries are to be submitted via the ECPS website. Full proposals are by invitation only. Those seeking an OT award are to submit their full proposal via the ECPS website. Those seeking a cooperative agreement award are to submit their full proposal via the grants.gov website. The summary of proposal and detailed proposal should not exceed 30 pages, and this excludes the statement of work. The evaluation selection, each conforming solicitation summary will be evaluated against the criterion 1, 2, and 3, and full proposals will be evaluated against criterion 1 through 4 in descending order of importance. Selection for award will be made as outlined within the ISO. The evaluation criteria. The first evaluation criterion is the overall scientific and technical merit, and this is applicable to, once again, the solution summary and full proposal. The second criterion is the proposer's capability or related experience. This is applicable to the solution summary and full proposal. The third criterion is the potential contribution and relevance to the ARPA-H mission. Once again, this is applicable to the solution summary and full proposal. The last criterion is reasonableness, realism, and affordability. And this is the only criterion that's only applicable to the full proposal. Final guidance. We ask that you monitor SAMS.gov and grants.gov. Any and all changes to the ISO will be made via formal amendment and posted to the SAMS.gov website. No information discussed at today's Proposers Day should be construed as modifying the terms and conditions of the ISO. We ask that you conform to all the ISO requirements, thoroughly read the ISO, pay special close attention to the eligibility requirements. Non-conforming proposals may not be evaluated or considered for award. Some of the reminders we have listed here, including the SF 424, research and related key personnel profile, research and related personal data, and the project abstract summary of some of the documents that we typically see missing with cooperative agreements, which is why we have this reminder here. These are not applicable to other transactions. Some important dates and deadlines to consider are the solution summary is due May 28th at 9 a.m. The full proposal is due August 20th at 5 p.m. If you have any questions, we ask that you submit them via the print mailbox at print@arpa-h.gov. And one additional guidance is that we ask that you frequently review the frequently asked questions website, read the ISO, read the frequently asked questions, and if you still have questions after that, we ask that you submit them once again to the print mailbox. We ask that you write your questions as clearly as possible. And this next one is extremely important. Please do not include proprietary information via email. And in some cases, ARPA-H may rephrase questions or consolidate similar questions for administrative purposes. And if there is a contradiction between the frequently asked questions and the ISO, we ask that you defer to the ISO. And this ends my portion of the contractual side of the ISO. Next, I'll turn it over to Emily, who will go over some of the financial questions of the proposal.
Thank you. Hello, it's a pleasure to have you here today. My name is Emily Cortese, and I am the Business Financial Manager for Print. I'll be working closely with you and your team to ensure that you're receiving funds and spending funds accordingly. But today I'm going to briefly talk about some of the best practices when writing your cost proposal. Earlier in the presentation, you saw the timeline for the print program. And so you can already see that in order to get on contract quickly, we'll be asking a lot of you in a short amount of time. You've seen the expectations for the solution summaries. We're hoping to review them and turn them around quickly, maybe in about a couple of weeks, so that we can let you know who we're inviting to write full proposals. As far as the budget for the solution summary, the print team is requesting just a rough order of magnitude and a timeline for the federal funds requested. This includes a breakdown of work by direct labor, labor rates, subcontracts, materials, equipment, other direct costs, and indirect costs, profit and cost sharing if applicable. With proposals coming in late August, items 1 through 7 on the right-hand side of the slide will be taking place throughout September and October, and then contract negotiations with your agreements officer will take a little bit longer. You've seen this slide already. Cooperative agreements and other transactions are the two types of awards we're expecting for the solicitation. Depending upon what sort of agreement you prefer will require a slightly different variations in terms of the forms that are required for each. You'll notice that the final ISO has not been posted yet. It's expected that that will be posted prior to the solution summary deadline. But once it is published, you'll also see some additional documentation, including specific cost proposal spreadsheets for each of these types of agreements to include in your final proposal. Please keep that in mind when you're looking at all of the final documentation on sam.gov. Here are some do's and don'ts and some common reminders to make your cost proposals the best that they can be. They'll ultimately show the ARPA-H team how much funding you will need and spend throughout the life of the program. Putting effort in on these spreadsheets will significantly reduce the burden on the team to review the cost proposal and reduce the number of times we have to come back to you and ask follow-up questions and additional documentation for updating your proposal. Some of the most important things here that I'll highlight is the task cost breakout by fiscal year and phase. That's very important. It's not just a breakout by fiscal year, but also by task and subtasks. And if you can do it even by month for all of the phases of the project. Please provide any supporting documentation for an item that's more than $5,000. That could be a single item that's $5,000 or 100 items that cost $50 each. For anything over $5,000, we'll need you to provide some sort of quote that justifies the expense. An itemization of all subcontracts. Our agreements officers would agree. Document everything, please. If you have letters, statements of work, proposals from potential subcontractors or consultants, please include them. And make sure that you include in your proposal how those costs are realistic and appropriate for the technical and management approach you've laid out in your statement of work. Please don't forget to utilize the ARPA-H cost proposal checklist. Please don't change the formulas in the spreadsheet. Please don't cost strategize. Ask for what you need. Don't think that there's a low cost or a high cost that's going to make your proposal more favorable. We expect that you're going to request exactly what you'll need to get the work done. And one thing that's not listed here is to please use the 2024 NIH salary cap in your figures. It was just updated to $221,900. We'll be asking for justification for all labor costs as well. And then lastly, here's the cost proposal checklist that I mentioned previously. So you'll have that to review at your leisure as well, but just to emphasize, please document everything. And now we'll take a quick break and meet back for the presentation from our patio office. Thank you.
Next, I'll invite Chan Siv to give an intro into patio and some of the government resources we can tap into.
Hi, everyone. Thanks for being here. So my name is Chan Siv. I'm a technical CETA within the Health Science Futures Mission Office, as well as the Patio Liaison. So today I'm going to talk to you a bit about the Patio Office and how they've been working with the program managers and eventual performers at ARPA-H to think about transitioning of the technology and solutions. As Amy and Ryan had mentioned earlier in this meeting, there's, transition is a huge component of how we are going to get technologies and solutions to the people. So to get a better understanding of how PATIO, or Project Accelerator Transition Innovation Office, fits into ARPA-H, so if you remember that the mission for ARPA-H is to accelerate better health outcomes for everyone. So in order to, when we think about the technologies and solutions that are coming out of ARPA-H, we want to make sure that these solutions actually get to Americans. So PATIO was stood up at ARPA-H to increase that probability that ARPA-H funded health technologies will reach Americans by identifying barriers and providing transition and commercialization services to program managers and performers. So when we think of transition, at ARPA-H, like transition, it's something that you think about even at the beginning of a program. So for Ryan's print team, for example, Patio has been working with Ryan at the beginning of the conceptually as he starts to think about the program and how he starts to think about the metrics that are in the TAs, right? If you think about how these technologies are going to reach Americans, you're going to have to think about equity considerations. You're going to think about all the regulatory hurdles. As ARPA-H is a health agency, so there's a lot of different challenges that Ryan and his team needs to think about, even as he puts out this solicitation. So as mentioned earlier, ARPA-H will not fund performers in perpetuity, and that's why this office exists, so that way we think about the transition challenges really early on in the process to ensure that success. So as y'all may, y'all here are working on different technologies, it may be clear to you where your technologies will transition to. But if we think more broadly about all of the solutions coming out of ARPA-H, these are some of the typical transition partners, right? Depending on the stage of the technology, it's more obvious to transition to, a startup, or if it can be licensed to other large companies, but some of the other transition players here can be healthcare systems, government agencies, NGOs. So I think when you are writing your proposal, we encourage you to think about how your solutions are going to be transitioned to at the end of this, right? I think at ARPA-H, we want to. Ryan and his team will support the performers at Print to think about all the transition partners and think about, like, what are the things that that the team can do to prepare you for whether it's your upcoming meeting with regulatory agencies and other introductions. When the Patio office was stood up, they used something called the IDEO framework to think about how to structure the team. So the IDEO framework considers feasibility, viability, and desirability in thinking how innovations should be built. So when we look at those things, these are the different divisions within Patio. So the first division, led by Jenica Patterson, that is the Technology Transition Office. So the aim of this office is to build that transition-ready program by de-risking solutions from program design through performance. So I'm going to talk more deeply about the capabilities within this office in the next slide, but I just want to say that, transition is hard. There's so many components to think about. The market has to be right. There are IP challenges, there are funding challenges, there are many reasons why, a technology may not be able to transition. And some of these due diligence happens within the T3X team. The next team, when we think about the desirability piece of making something successful, the health ecosystem and engagements team, or HEAT, helps with some of that. So the aim here is to create meaningful bi-directional communication between ARPA-H and America. So how ARPA-H does this is ARPA-H participates in a lot of symposiums, conferences, speaking at universities. But the questions that are asked at ARPA-H are questions that are of interest, by the community. So it's we want to go. ARPA-H goes out into the community to talk with many folks to understand what are those hard challenges and bring them back and form new programs and initiatives at ARPA-H. And the last piece here, which Amy briefly mentioned about, is the ARPANET-H. So if you all may have heard about the hub and spoke model at ARPA-H, but the goal here is to, reach all Americans. So how do we? So in order to do that, we need to be where they are, right? So the aim here is to connect this fragmented health ecosystem through projects, events, and democratized learning. So within ARPA-H, there are 3, I would say, geographic locations. So the DC office is the stakeholder and operations hub. That's where we connect with a lot of folks at other government agencies. But we also have 2 other locations. One is the Investor Catalyst Hub in the Boston area, and one is the Customer Experience Hub in Dallas, Texas. So the purpose of these hubs is to ensure that ARPA-H is taking that learning from experts within the field, academics, nonprofits, everyone, and taking those learnings back to ARPA-H to make a better program. So you may have been involved in some of these network activation calls that were recently put out by some of these hubs, but these spoke members make up a very diverse network, and we realized that in order to develop the best solution, you need to incorporate all of the experts here in the U.S. So if you do get funded by an ARPA-H, you'll likely interact with these 3 divisions and the people listed on here to help think through, some of the transition challenges. So to dive deeper into one of the services that that worked really closely with Ryan is here for T3X. Again, the aim was to build transition-ready programs by de-risking solutions. So some of the ways that we worked with the print team is something called an experts in residence program. So when program managers enter ARPA-H think about a new program, they're usually matched with an expert in residence. What this means is these are folks from the commercial entity as well as other industries who are really well known, recognized for transition and their commercialization expertise in identifying identifying blind spots that the program manager and the teams might not have considered. So for example, print is a really novel area. So there may be things that, that Ryan and his team has not considered, and this is where the expert comes along. During the award and performance period, there may be a need to identify bespoke regulatory and reimbursement support. So Ryan mentioned that, after phase 1, I believe that, there's a possibility to have an interact meeting with the FDA. And what we can do at this stage is to bring in a regulatory expert and Ryan to help Ryan and the performers think through all of the requirements that should be considered so that you go into this meeting prepared. The third thing here is the due diligence and landscape analysis. So before print was launched, the team performed a market analysis and landscape analysis for the print team to identify, what is the market, who are the competitions, what are some of the metrics to consider. So all of this is important at the beginning of the program so that you build out a program to reflect that and transition ready. We realized that, at the end of the program, the market dynamics may have completely shifted and there may be 20 different competitors now in the market. And that is something that, Ryan can continue getting support from Patio on to identify those key players and connect performers to whoever that's needed to help transition this out. And then the last thing that this team manages is the small business program. So this is congressionally mandated program where it allows ARPA-H to provide funding to small businesses to use innovative approaches to enable certain advancements in technology systems and platforms. So you may have participated in some of the solicitations previously for the small business program. So how this works is the program managers are the ones who are putting out the ideas. So the ideas may be complementary to the print program or can be complementary to, Brian's entire portfolio. So this is something that ARPA-H will continue doing. So in the interest of time, I'm going to stop there. I think, as I mentioned, there's a lot of services at Patio, and the way that as a performers at ARPA-H, how you would interact with Patio is through your program manager. And I think throughout the different phases, the program manager can initiate some of these support services on your behalf to help you be more successful in transitioning at the end of the program. Thank you for your time.
Next, I'll have Tammy Selleck give an overview of animal and human subject research.
Hi, everybody. I'm just going to do a quick overview of some of the requirements, extra requirements that are put in place for protection of our animals and human subjects in the course of research. First, we're going to talk about animals, and then we'll go into the humans. A lot of the requirements are similar, but there are some that are they are unique. There is a clear definition of what's an animal. An animal is it's a living vertebrate. Or if it is dead, it's killed for the purpose of the research. It's a larval fish or amphibian. You can read all of these right here. What's important is that what is not an animal is an unhatched egg, an invertebrate, and any kind of portion or whole of an animal not killed for research. So things that you might find in a grocery store, obviously, they're not considered animals or any part and parcel. So it's very clearly defined what is an animal and what would require these additional documentation and steps to be included in your proposal. There are some overarching guidelines, and these are going to be provided to you as I understand it. So you can click on these links for additional information. They're both, there are two guides. One is for the overarching animal welfare, and then the guide for care and use of laboratory animals. These are NIH prepared documents and overseen by the OLA, which is, I can't even remember what that acronym stands for. It's Office of Legislative Animal Welfare, I believe. Okay, so award policies. Institutions engaged in animal subjects research must have the following in place prior to any animal research to begin. This is multiple documentation requirements. It is a current IACUC approved protocol, and that is institutional Animal Care and Use Committee, and it is a part of the organization conducting the research. They'll have an independent review of the protocol. Basically, protocol is a step-by-step instruction of the experiments that you're going to conduct using the animals. A valid assurance of the institution. So you have to have the institution itself has to have a valid assurance. These documents are in, you can see examples of them and more descriptors when you click on the links on the previous slide. A vertebrate animal section completed. That's a document that you just need to complete with certain relevant information about your experiment and the animal. And then the interinstitutional assurance if applicable. All of these documentation, we have resources in-house at ARPA-H to be able to do a review of these documents for completeness and accuracy. And all of these have to be approved prior to any research being conducted that involves the animals. We do have something that we can put in place, which is a restricted award, where you can start doing other sections of the statement of work, and you can start work without having those assurances and approvals in place while you continue to get your packages together and get approval for those. There are certain situations that will require a re-review of your protocol and documentation, and one of those is a protocol amendment. And not all amendments will require, there's some that have expedited review and then others require an entire re-review of the package. And the circumstances are outlined in the the bullet points there. So what my point is that be careful. So when you have changes to the inside the lab room number, the PI that's in charge, or method of anesthetic administration, there are certain steps that are going to require you to rewrite and resubmit for approval. IACUC approvals are good for 3 years, or the length of the award, whichever is shorter. So 18-month contracts, obviously, would be just one. And at the end, at the 3-year mark, you have to go through the evaluation and approval process. Non-compliance. We certainly don't want any of these. Non-compliance is when we fall short of meeting any of the requirements imposed by the legislation. And it can lead to cancellation of the award in extreme cases. We do, if you have any questions, there'll be an email address provided at the end of this where you can reach out directly with any specific questions. And then you'll also be able to reach out to Ryan's team to get other questions answered as well. So we do have resources available for you for anybody that's unfamiliar with going through this process. On to human subjects research. Definition of a human is applied to all of the research efforts, but it's a living individual. About an investigator, whether professional or student, conducting the research, obtains information, interacts with the human to gain information to supplement the research. Very specific, but for this program, it's going to be very clear. There are certain situations where that definition can be interpreted in different ways. And again, using the resources that we have available to make sure that we're falling inside that, understanding the clear lines of what is considered HSR. This one's going to be pretty clear. Human subjects research regulations, just like in animal subjects research, there are regulations in place. It's a 45 CFR Part 46. And this is what we call the common rule. And most federal organizations have adopted this as the way to conduct business and the appropriate ways to protect our human subjects. There are certain subparts that require additional protections, such as pregnant women, children, prisoners, and military service members. And so for any of those populations, they would require additional protections and additional documentation requirements. So just like animal use, all institutions that are involved, this includes subcontractors. This is very, very important. It's not just for the prime awardee. All subcontractors have to have these same approvals in place prior to engaging with humans. But a current IRB approved protocol, an IRB, just like that institutional animal care and use committee, is an institutional review board. And it's composed of 5 members of various different backgrounds and education. And they do an independent analysis of your protocols to determine if it's, where to improve or change or approve your protocol. And you'll also have a valid assurance for human subject research as well. All of these, just like your animal subjects, these packages need to be approved prior to any HSR-related activities being started on the award. And again, we can do restricted awards for you to begin the non-human subject-related tasks. Your package for human subjects research is going to include at minimum your approved full protocol and an example of your informed consent documentation, which is basically for any of your participants to make sure that they understand the experiment that they're about to participate in, and that signature on there. It could also include any combination of the items listed there on the bottom. Definitely going to have proof of training for your PI and other participants on the team. Your data collection materials, if there's any kind of information, survey, or any kind of information gathering materials. The CV of the PI and other participants, and recruitment and advertising materials. Okay, only the IRB, they have the authority to approve your protocol, and they're the only ones that have that authority to be able to give you that stamp of approval to move forward. There are certain things that are going to be deemed exempt research, but it's still considered HSR. I don't think any of the protocols that are going to be part of the program are going to fall within that. category, but it is just FYI. Research considered minimal risk and not greater than minimal risk, or it's basically a determination of the risk to the participant based on a baseline of everyday life. So if it's a higher risk or a lower risk, will impact timeline to approval, and it's also going to be spelled out in your letter from your IRB. It will tell you what level of risk is posed to the participants. Just like animal use, any amendments to your protocols, any changes to the PI, any changes to your protocol or experiments is going to potentially require resubmission and reapproval. Again, reach out to our resources if you have any questions about what changes, if you think that you'd like to make, if that would require a resubmission of your protocol package. And that's it. Any questions? We're not taking questions. All right. Yep. That's all I got. Thanks, everybody.
Next, I'll invite Dr. Yingfei Xu to give an overview of some frequently asked questions that we've been receiving to kind of clarify some issues for everyone. Thank you.
All right, thank you, Ryan. So first of all, I would like to point out two things. First, this is a informational only session, so we are not taking live Q&A. Second is the ISO is really has the highest authority. So if the final ISO if you find discrepancy between the slide stack and the final ISO, ISO will basically have the higher authority and supersede that. So, that. A lot of questions we received so far is on the teaming. We got a question asking whether the proposer is the university or each individual PIs, and whether there's limitation on the number of applications you can put through. So currently you can see the draft ISO, you will see we have a paragraph describing you can only be one prime and one sub. In the final ISO, that line will be removed. So there is basically no limitations in the number of proposals or the solution summaries you can put forward. But bear in mind that you are basically competing yourself, because the print selection process is a competitive process. So you don't really want to be in that situation. Second, what kind of organizations that we accept? That's basically any. We don't treat one differently from another. So it can be a mixture of any, it can be industry, academia, anything. Is there a limit to how many institutions can team? Not really, but we really want to be reasonable, so that the teaming members should be the experts of one or more specific area that are essential to the completion of your project. So it's really, we don't have a limitation on that. Some more. Can we have multiple PI? No, there will be one PI and others will be co-Is. Can we change team pathway through the program? We understand this. It's a 5-year program, and then different situation may arise. This will be dealt by a case-by-case basis. Well-established team for us. We are not really doing matchmaking, so we cannot force you to our collaborators. We do have the site, the teaming profile site. You can submit your 200 words entry. We will post it, and then you will have other parties coming to you. The matrix. The matrix seems aggressive. Do you really intend for us to hit this matrix? We do really want you to hit this matrix. We understand the expectations are high, but also that's how the overall ARPA-H standard is. So we do hope that at the end of the program, you will hit this matrix. What if you don't hit one? Will you stop? That's in the program, we have two phases. First phase up until three years, yes. And then at that point, we will have a go or no go decision. Matrix is definitely one of the criteria that we will be using to judge everybody's work at that point. You already have something that you might have go faster. Of course, do it. We genuinely hope that's the case. Do you have any preferred technological approaches? You will have the full freedom to complete the project as long as you can fit in the matrix and the milestones, eventually being listed in the ISO. What if there's other matrix? This, I think it's again, a case-by-case basis. We understand the technology evolves. In the later time, there might be new, better matrix to gauge the success. We will be willing to look at those with you. Can we meet to discuss ideas? We can answer questions, but really we cannot prescribe the approaches to you. We can also really tell you whether whether we like or not. If you feel like we can tell you whether your approaches is within the scope or not, but really we do not have preference of your approaches. If you think it's out of scope of print, you can also propose to the open ISO that's currently in 4 different offices, as Amy mentioned. International proposers, we get a lot of questions on this as well. So basically, we do not prohibit international proposers. They can be your co-Is, subcontracts. There are a few caveats for international proposers to collaborate with US entities. We will work with you on this type of situation. Government proposers, federal entities and federal sponsored entities such as FFRDC, UARC are not eligible under this call, unfortunately, not as prime or sub-awards, but the PM is willing to work with government partners to work on ARPA-H missions and projects related to his interests. How big can the program be? What's the overall budget? So the main takeaway is we don't prescribe you a number. There's no budget ceilings, but do bear in mind that whatever you propose should be reasonable, and more importantly, it should be associated with your approach. So propose something which you think reasonable and fit for your technical approaches. Can you explain the rules around the salary cap in your cost proposal? So there is government regulation and restriction on that that has been very clear. you're welcome to read this. IPs. This is also a hot topic. Who owns the IP? In general, the government will not take ownership of the IP generated under the awards. Under cooperative agreements, you will retain the ownership of IP, but the government will review it for federal purposes. For OT, there's more flexibilities. It will be undergo in the negotiation process. So we do want to, we basically do not want IP to be hindering you from the technical advances. Okay, solution summary feedback. So deadline for the solution summary submission is May 28th, right after the Memorial Day weekend. The solution summary will be reviewed based on the criteria 1, 2, and 3 as put forward earlier. We will try to give you a final decision, a binary final decision, whether to invite or not invite. But in the process, it's possible that we contact you for additional information. But the final decision will be whether you are invited or not invited for a full proposal. We do expect this decision will be made a few weeks after the deadline. So it will be sometime in June. After the solution summary, it will be the full proposal. The full proposal deadline, again, is August 20th. So that should give you enough time, even after you receive the feedback from the solution summary decision. So we will only evaluate the full proposals that we have already invited. Will feedback be provided for full proposal? Yes, we will provide the feedback. You can contact Print Mailbox for scheduling a feedback call. That's also allowed. Then dive into some technical questions. Is GLP, GMP manufacturing required for the small and large animal studies? As Ryan mentioned, yes, we will need GLP and GMP manufacturing capability to be used in TA 1 and 2 so that these cells can be used later on in the program. Bioink and bioreactor required product deliverables. The required deliverable of the program is the cell manufacturing and the restoring of the organ functions. We understand that these 2 components are critical to the completion of these goals. You don't necessarily need to create a new biomanufacturing infrastructure. Existing manufacturers infrastructures can be leveraged. And in TA1, you can see Bioink may serve as one approach. A biobank may serve as one approach to generate necessary cell types. But in TA2, you are expected to develop a master cell bank under GMP requirements as a deliverable. Are there any restrictions on what type of cells or animals that can be used? For the cell source, the performers will have the freedom to choose. We understand many of you already have an idea or prior experience in working with one type of cells. So there's really no restriction for animal testing. We do want you to use humanized small or large animals for this program. Can inorganic products be used to achieve the required end results, such as artificial organs or bionic organs? The short answer to that is, as long as you hit all 3 TAs, outlined in the ISO, we will accept that. But then if your proposal do not have one or two TAs, which means you don't have the cellular components to it, it will not likely fit within the print program. In that case, it will go to the more open call ISO. Again, one thing I cannot stress enough is read the ISO. We have people dedicating to the mailbox and will respond to you. That's it.
So we're going to edit our schedule a little bit, given that we're so far ahead. We thought, as much as we said we weren't going to entertain questions at this time, since we have time to, we'll open it up just so, to make the best use of everyone's time. So at this time, anyone in the room, if you have anything you want to ask, feel free to shoot.
No, there's no mandated cost sharing. The question was regarding the salary cap and if there's a cost sharing mandate, and no, there's not a cost sharing mandate. Yes, so the question was regarding the budget. If there is a budget that needs to be submitted for each phase, then yes, one has to be submitted for each phase.
Yeah. So we're still going to work on try to lock that down. We're still in the phase, so we haven't committed to a particular organization yet. But this will be mostly for the cellular components of this. So yeah, nobody's going to be like handling your organ per se at this point. It's kind of more on kind of the TA1, TA2 aspects of are the cells safe and manufacturable. Yeah. Yeah. So the question is, where the organ costs come from? So I think the main basis of that was kind of looking at the manufacturing costs of. So basically, like depending on the organ, estimating about how many cells we think were necessary, likely to restore function, and then kind of basing that off of published costs and talking to other experts. Surely they're educated estimates. And, they're not hard numbers. They're kind of based on everything we could collect at the time and based on the early stage of development.
So no, you'll have an opportunity to put your team together by the full proposal due date. So there is an expectation that a team would be developed and submitted with your full proposal.
Yeah. We're expecting that you're going to be printing cells as part of your solution.
Any other questions?
Yeah, basically, I think, so the question is, how do we anticipate the performers interacting with selected CRO, and basically just ensuring that, all your intellectual properties intact, but also making sure that that they can test whatever you're developing the process. You would share kind of, can they recreate whatever processes that you're having, that you developed as part of the program. And it'd be basically on the cellular, so like TA1 and 2 portions essentially. Because we wanted to make sure that whatever data you put forth, that we can be able to reproduce that, essentially a third party to be able to do that. Yes, so we're not prescribing an exact timing because it may vary between teams, but basically once you have a process, you're like, okay, we think this looks like it's working. Here's the data. Then we'd want to take it and be like, okay, can we reproduce this? Yeah, exactly. What was their additional pay? Well, I guess, certainly it'll be probably a little bit of a case by case, and understanding your exact solution. But basically, as long as the solution is going to print the cells at some point and they'll be part of, in creating that organ and maturing that organ, that solution would be acceptable, if I understand correctly. Yeah. So we don't say you necessarily have to print exactly at this timing. If there's some reason to, stagger, or you have some other scaffolding or something that you're considering.
So it looks like we have no other questions, but we do want to remind you that we do have that print inbox. So if you think of any other questions after today, just make sure you submit that email. Thank you.